Dear students, this is our 17th lecture on medicinal chemistry and third lecture on antiviral agents. Uh, in the first two lectures, in first lecture, we have discussed about what are viruses and what are the commonly used antiviral agents in addition to the virus replications, how uh, it take place in a host cell. Then we have also discussed about the common type of viruses like HIV, human immunodeficiency virus, herpes simplex virus, hepatitis B virus, hepatitis C virus, then varicella zoster virus, then respiratory syncytial virus, influenza A virus, and influenza B virus. So these are some of the common viruses which are very common in case of humans in these days, and the drugs which are used to control these viruses, they are called as antiviral drugs. And in the second lecture, we have discussed about anti-HIV agents and replication cycle of HIV virus. In addition, we have also discussed about the synthesis of HIV protease inhibitors. Now, as you know, that to control the HIV replicative cycle, we can use these five categories of antiviral compounds as drugs. One is nucleoside reverse transcriptase inhibitor, nucleotide reverse transcriptase inhibitors, non-nucleoside reverse transcriptase inhibitors, and HIV protease inhibitors and fusion inhibitors. In the last lecture, we have already discussed about the synthesis of some of the commonly used HIV protease inhibitors. And in this lecture, we will discuss about nucleoside reverse transcriptase inhibitors and nucleotide reverse transcriptase inhibitors. Whereas in the next lecture, we will discuss about these non-nucleoside reverse transcriptase inhibitors. Now, first is lamivudine. This is the IUPAC name of lamivudine, and this lamivudine is basically a nucleoside reverse transcriptase inhibitor, and this is a kind of deoxycytidine analog. And this antiviral agent is used to inhibit the reproduction of HIV virus and hepatitis B virus. And this is effective against both of these viruses, HIV as well as HBV, that is hepatitis B virus. As you can see here that this antiviral drug lamivudine is having two chiral centers. One is this one and second is this one. So therefore, because of the presence of these two chiral centers, it has four stereoisomers. But all the four stereoisomers, they are not equally active. And out of these four stereoisomers, this is called as 2R cis isomer, 2R5S, or this is 2R cis isomer. This is the most potent or most active against HIV and HPV. So we can say that this isomer is having anti-HIV and anti-HBV activities. And the toxicity of this isomer on some cell is lower than its enantiomer or racemic body. As far as the original synthesis of this lamivudine is concerned, this was developed at GSK Laboratories, and a patent was filed in 2000 by Goodyear and et al. What they have used as substrate is this compound L-methyl glyoxylate, which is formed from glyoxylaldehyde and menthol. They condense with each other to give us this ester aldehyde. When this ester aldehyde, L-methyl glyoxylate, reacts with dithienediol, which is a dimer of this thiol acetaldehyde. In presence of toluene, this undergoes reaction at this aldehydic center and this thioacetaldehyde. So basically what happens is this sulfur, because of the lone pair present on this, act as a nucleophile and attack the carbon of the carbonyl, and then this oxygen will acquire negative charge and this will attack this electrophilic carbon, and this will go as O negative, and this will take up the proton from here to here, and this is how the cyclization will take place to give us these types of esters having 1,3-oxathiolane ring. So these optically active 1,3-oxathiolane esters on reaction with methane sulfonic acid in dichloromethane and DMF in presence of thionyl chloride, we know nucleophilic substitution reaction alcohols, they are converted into the corresponding alkyl halides when they are reacted with thionyl chloride. So the corresponding chloride of 1,3-oxathiolane 2-carboxylic acid ester will be formed. Then this 1,3-oxathiolane derivative of having this chloride ion reacts with silyl cytosine, which is synthesized from cytosine. So cytosine on reaction with hexamethyl disilazane, this silyl methylation of this amino group, and this oxygen will take place and we will get silyl cytosine, and this silyl cytosine will react with this 1,3-oxathiolane chloride. So basically this nitrogen will act as a nucleophile and this will attack this carbon and halogen will be removed, and we will get this type of pyrimidine 1,3-oxathiolane derivative is formed. And then when this derivative is reduced with sodium borohydride in aqueous medium in presence of sodium hydroxide, basically what will happen first, this ester will be converted into CH2 group, and then hydrolysis of this will take place and the corresponding alcohol will be formed. So this is how we can synthesize lamivudine from L-methyl glycoxylate. Then another method is which have been given recently in 2020 only by uh, David Snyder, and they have used a different substrate, that is butene dioate. So this diester is used, uh, and this diester condenses with the same substrate which was used in the first method, dithienediol, which I have told you is a dimer of this thiol acetaldehyde. So this thiol acetaldehyde condenses and cyclizes with this dimethyl dioate, and we get the same type of 1,3-oxathiolane derivative as the product through cyclization, nucleophilic substitution and cyclization lead to the formation of this heterocyclic compound. Then this ester reacts with this moiety, which is synthesized from 2-chloropropanic acid on reaction with this base sodium methoxide. So methoxide will act as a nucleophile attack over here, replace chloride from here, and then sodium ion will get attached to the sodium salt of the carboxylic acid will be formed. Then this will condense with this 1,3-oxathiolane. Basically, this acid part will react with alcoholic part, and the corresponding ester will be formed. Then bromination of this ester followed by reaction with cytosine. So first this will hydrolyze, and then this will add to cytosine will add to this moiety. Basically, if you see here, we have used pavallyl chloride and this tertiamine, levamisole hydrochloride. This tertiamine is used for the resolution of this racemic mixture into the optically active enantiomer, which is required for the synthesis of lemopodine. So after the condensation, we will get this type of cytosine derivative, and this cytosine derivative on reduction and hydrolysis will lead to the formation of lemopodine. Then next antiviral drug is retrovir, and its brand name is zidovudine, and earlier it was known as azidothymidine. Uh, Commonly nowadays also this is called as AZT or azidothymidine. Uh, Again, this drug is active against HIV, and this is a nucleoside analog reverse transcriptase inhibitor. And chemical name is 3-azido 3-deoxythymidine. So 3-azido 3-dioxythymidine as the HIV anti-HIV agent. The uses of this drug, this is recommended for use in combination with other antiretrovirals, and this is used to prevent mother to child spread during birth and needle stick injury or other potential exposure to HIV disease. Side effects, common side effects are headache, fever, and nausea, whereas serious side effects may be liver problems, muscle damage, and high blood lactate levels in the patients. It is safe in pregnancy and can be used very commonly. The mechanism of action of this drug is that it will inhibit the HIV's reverse transcriptase enzyme. As you know that reverse transcriptase enzyme is used for synthesis of DNA from the viral RNA. So if RNA of the virus is to be converted into viral DNA, then this reverse transcriptase enzyme is necessary. And this AZT, what this will do, this will inhibit this enzyme, and that is why there will be no replication of RNA to DNA. This antiviral agent was first described in 1964 and it was approved in 1987 for treatment of HIV, and it is in the list of WHO list of essential medicine, and this is also available as a generic medicine in all the countries. Now, this can be synthesized retroviral zidovudine can be synthesized from this substrate. This is 1,2-deoxy 5-O-tretyl beta D lioxylthiamine. When this is reacted with methane sulfonyl chloride, the corresponding mesylate at this hydroxyl function group is formed. Then this mesylate on reaction with lithium azide will convert it into the corresponding azide. So this azide derivative uh, when with inverted configuration at C3 of the pyrronyl ring, when this is heated with 80% acetic acid, the hydrolysis of this trityl protection take place, and we will get zidovudine or retrovir as the product. So this is how zidovudine can be synthesized from this thiamine derivative. And next is didanosine. This is sold under brand name Videx. Again, this is a reverse transcriptase inhibitor used as anti-HIV or anti-AIDS drug. This was first described in 1975 and approved in 1991 as anti-HIV agent. This didanosine is nucleoside analog of adenosine, and it differs from other nucleoside analogs because it does not have any of the regular bases which are present in the nucleosides. So this has hypoxanthine attached to a sugar ring. So here within the cell, didanosine is phosphorylated at this hydroxyl function group to the active metabolite of dideoxyadenosine triphosphate by cellular enzymes. So, like other anti-HIV nucleoside analogs, it also acts as a chain terminator by incorporation and inhibits viral reverse transcriptase enzyme by competing with natural ATP. The common adverse effects or side effects, they are diarrhea, nausea, vomiting, abdominal pain, fever, headache, and rashes. Whereas pancreatitis is rarely observed but has caused occasional fatalities. Then other serious adverse events, they are retinal changes, optic neuritis, and then some effects on the liver functions. So in 2010, US FDA they have issued a statement that the patient using didanosine, they are at risk for a rare but potentially fatal liver disorder, which is called as non-creatic portal hypertension. So therefore, this use of this drug has been limited. Now, didanosine or Videx can be synthesized from L-glutamic acid. So, when L-glutamic acid is reacted with NaNO2HCl, basically what happens is we know that NaNO2HCl will react with the primary amines and the corresponding diazonium uh, salt will be formed, and then this carboxylic acid will attack this carbon and deamination will take place. So basically, this is a stereospecific deamination lactonization of the glutamic acid, which take place, and we will get this type of gamma butyrolactone acid. Then this gamma lactone acid is reacted with ethyl alcohol in presence of paratoluene sulfonic acid. We know alcohol will react with carboxylic acid to give us the corresponding esters. Then this gamma butyrolactone ester reacts with sodium borohydride. So basically, this reduction and hydrolysis will take place, and we will get the corresponding alcohol will be formed. And this hydroxymethyl gamma butyrolactone, which is optically active S gamma hydroxymethyl gamma butyrolactane, is formed. This is basically, we can say, is the starting material for many types of other antiviral compounds, which you will see in the subsequent slides also. Now, this S gamma hydroxymethyl gamma butyrolactone on reaction with the benzoyl chloride, benzoylation will take place. Benzoyl ester will be formed again. Then this benzoyl ester will react with diisoamyl borane, hydroboration, oxidation of this ketonic group will take place. This ketone will be reduced to hydroxyl group through hydroboration reaction. Then this will again react with acetic anhydride in pyridine, and the corresponding ester of this alcohol will be formed. And then this will react with trimethyl bromide. So bromination at this carbon will take place with the removal of acetate ion from here. So trimethyl acetate will be formed, and this bromide will be shifted to this carbon. The corresponding bromide will be formed. Then this furane bromide, tetrahydrofurane bromide derivative will react with tetrahydrofuryl bromide, will condense with silylated adenine. This is silylated adenine, and this nitrogen trimethyl bromide will be removed, and this carbon will get attached to this nitrogen. So this is how we will get the compound. In this compound, where coupling of this carbon with this nitrogen with a new carbon nitrogen bond is formed. Then this uh, benzoyl ester part will be hydrolyzed in the presence of ammonia and methanol to give us again the hydroxyl functional group, and then deamination of this will take place with ADA enzyme, and this will be converted into ketone. So this is how we can convert L-glutamic acid to adenosine. The next is pivate or gelcitabine. This is also called as 2-3-dideoxycitidine. This is also commonly called as dideoxycitidine. Again, this is antiretroviral drug used for treatment of HIV and AIDS. HIVID is its trade name, and again this is a nucleoside analog reverse transcriptase inhibitor. The adverse effects of this are nausea, headache, like the other nucleoside analogs we have seen. Then more serious adverse effects are peripheral neuropathy, which can take place in one third of the patients. with advanced diseases, then oral ulcers may be there, then esophageal ulcers may also be there, and rarely we can have pancreatitis also in some of the patients. Mechanism of action is same as we have seen in case of other analogs of nucleosides. Here, this is a derivative analog of pyrimidine. So, it is derived from deoxycytidine by replacing the hydroxyl group over here by hydrogen. And in the T cells, it is phosphorylated and other HIV target cell into active triphosphate form. So here we will get an active triphosphate form in the T cells. Then this works as a substrate for HIV reverse transcriptase. Therefore, the chain elongation will be terminated or inhibited. The activity of reverse transcriptase enzyme will be inhibited. The mode of synthesis is the starting material is 2-deoxycytidine, this one. When this is reacted with benzoic anhydride in DMF, the selective N-benzoylation will take place, whereas there will be no effect on these primary hydroxyl group or secondary hydroxyl group. So N-benzoylation will take place. When this N-benzoylated deoxycytidine is reacted with pavyloyl chloride in presence of pyridine at minus 10 degrees centigrade, then the corresponding ester at this primary hydroxyl function group will take place. So pavyloyl ester will be formed. And in the next step, this reacts with methane sulfonyl chloride. So here mesylate of this hydroxyl group will be formed. And uh, this methylated pivolyl and benzoyl derivative under strong basic conditions in presence of sodium methoxide and DMF at minus 10 degrees centigrade. Here oxidation will take place. This hydrogen from here and methane sulfonate from here. Methane sulfonic acid will be removed. This will become carbon-carbon double bond, and hydrolysis of this pavylyl ester will also take place, and we will get the corresponding primary alcoholic function group. And then this compound is reacted with anion exchange resin, amberlite IRA 400. This will exchange uh, OH negative ions. So hydrolysis of this amide will take place in presence of this anion exchange reason, and amino group will be released. And after this reduction of this carbon-carbon double bond take place in presence of hydrogen, in presence of palladium on barium carbonate. 5% palladium on barium carbonate is used for hydrogenation of this carbon-carbon double bond, and we will get our drug, that is gemcitabine, as the product. The next is Virid. Uh, Virid is the trade name, and this is tenofovir diisoproxil. This is a drug used to treat chronic hepatitis B and HIV/AIDS. This is used with other antiretrovirals. So this is given as a therapy in combination with other anti-HIV or anti-hepatitis B agents. Common side effects, they are nausea, rashes, diarrhea, headache, pain, depression, weakness. All these are the common side effects of this drug, whereas severe uh, side effects they include high blood lactate, then enlarged liver may also be there in some patients. Then but it is safe during pregnancy. This is a nucleotide reverse transcriptase inhibitor, and this decreases the ability of the viruses to replicate. So conversion of RNA into DNA is diminished or decreased. This was patented in 1996 and approved in 2001, and this is again in the list of essential medicines of WHO. Uh, then mode of action is, as I've told you, that this is a nucleotide analog reverse transcriptase inhibitor. It selectively inhibits the viral reverse transcriptase enzyme in retroviruses such as HIV or other hepatitis B viruses. So again, it also uses DNA polymerases alpha, beta, and mitochondrial DNA polymerases gamma. This in vivo, this tenofovir diisopropyl is converted into tenofovir. That is basically hydrolysis of this isopropyl propyl group take place, and this is produced in vivo tenofovir, which is an acyclic analog of deoxyadenosine 5 monophosphate. This lacks a hydroxyl group in the position corresponding to 3 position. Then because of the lack of this will terminate the DNA chain elongation because of the absence of this hydroxyl. Then synthesis of this drug, that is tenofovir diisopropyl. This is synthesized from adenine. When adenine is reacted with this 1,3-dioxylane 2-ol derivative, optically active, R isomer, then ring opening of this will take place, and this adenosine at this will act as a base, and this will attack this carbonyl, and ring opening will take place, and we will get this type of amino propanol derivative. Then this amino propanol derivative reacts with this diethyl diethyl phosphate derivative, sulfone 4-methyl sulfonic acid ester in presence of a strong base like tertiary lithium. This will esterification will take place at this hydroxyl function group. This will act as a carbanion, and this will attack this hydroxyl functional group, and this part will be added to this hydroxyl function, and we will get this type of phosphonate derivative is formed. And then this phosphonate derivative reacts with trimethylsilyl chloride in presence of sodium bromide, and we will get the hydrolysis of this ester. We will get phosphonic acid derivative. So this phosphonic ester will be converted into phosphonic acid. Then this phosphonic acid derivative, that is tenofovir, which is produced in vivo, actually this reacts with chloromethyl propane 2 oil carbonate, and at these two hydroxyl function groups, these groups they will be attached, and we will get tenofovir diisopropyl as the product. So this is how tenofovir diisopropyl can be synthesized. The next is stovidine. The trade name is Zaret. This is again an antiretroviral drug, anti-HIV or anti-AIDS. Its function is this is again is used in the therapy in combination with other antiretrovirals, but this is not a first line treatment. It may be used for prevention after needle stick injury or other potential exposure has taken place. Common side effects like others headache, diarrhea, vomiting, rashes, and peripheral nerve problems. Then severe side effects may be high blood lactate and pancreatitis and enlarged liver, as we have seen in other antiviral drugs also. But it is different that it is not safe in the pregnancy. So this is a nucleoside analog reverse transcriptase inhibitor. Again, that means it will stop the or inhibit the replication of RNA to DNA by inhibiting the activity of reverse transcriptase enzyme. So this is a nucleoside analog of thymidine, and it is phosphorylated by cellular kinase. So this is thymidine. You can see if we dehydrate this at this place, this will be converted into stovidine. So stovidine triphosphate inhibits the HIV's reverse transcriptase. So here we will get the triphosphate derivative at this thing. So when the activity of reverse transcriptase enzyme is inhibited, then virus will not be able to make DNA copy of its RNA, and that is how this will act as a reverse transcriptase inhibitor. So there are two methods for the synthesis of stovidine. One is we can use this gamma beta-lactone alcohol, which was also used in some other anti-HIV agent previously. When this is reacted with this acid chloride, tertiary butyl acid chloride, basically ester of this hydroxyl group will be formed. Then reduction of this ketone with diol in THF will take place. This ketone will be converted into hydroxyl function group. Then when this is reacted with thionyl chloride and dichloromethane, this hydroxyl group will be converted into chloro group. So the corresponding halogen will be formed. And in the second step, when this is reacted with tertiary potassium hydroxide in THF, then dehydrohalogenation will take place. Hydrogen from here, Cl from here is removed, and we will get this type of unsaturation, carbon-carbon double bond is formed. Then this reacts with this base in presence of hexamethyl diacetylamide in trimethyl chloride and NCSTHF. Then basically this nitrogen will act as a base, and this will attack at this carbon. With the addition to this, then chloro will come from trimethyl chloride, and we will get this type of derivative. So when this is reacted under basic condition in the presence of strong base, this will be hydrolyzed. Sorry, strong base potassium tertiary butoxide THF dehydrohalogenation will take place to give you this carbon-carbon double bond, and sodium methoxide in methanol will hydrolyze this ester to this alcohol. So this is how strobilurin will be formed from this through this method. Then another method is that we can use tertiary butyl diphenyl silyl chloride and DMF. The corresponding silyl ester or silyl ether of this alcohol will be formed for the protection of this hydroxyl group. Then this will react with lithium diisopropylamide in THF with diphenyl sulfide in hexamethyl phosphoramide. Basically, what will happen? This will add S-phenyl group at this position. First, this lithium diisopropylamide, what this will do? This will create a carbanion at this position, so enolate will be formed. Then that carbanion will attack the diphenyl sulfide, and this SPH group will get attached, and this phenyl will be removed in the form of benzene. Then this S-phenyl derivative, alpha S-phenyl butyrolactone will react with the dibyl in toluene. Dibyl in toluene will convert this ketone into alcohol, that is hydroxyl group. Then that hydroxyl group will react with acetic anhydride in pyridine, and the corresponding acetate will be formed. Then this is reacted with this pyrimidine derivative, and in presence of SnCl4 and DCM, again the attack of this nitrogen at this carbon will take place. Sorry, this attack of this nitrogen at this carbon will take place with the removal of acetate ion, and we will get this type of pyrimidine derivative. And then this is oxidized with sodium periodate in water and THF mixture, followed by heating in presence of toluene and pyridine for the removal of this group, and hydrogen from here, and phenyl thiol will be removed. In hydrogen from here, PHS from here. This will create a carbon-carbon double bond in presence of sodium periodate, and heating in presence of toluene and pyridine will remove this protection group of silyl, and hydroxyl group will be generated there. So this is how this can be converted into strobilurin through this method. And then last is carbovir. Carbovir is again an anti-HIV drug, and this has low cytotoxicity, as was reported by Vinci and co-workers. Out of the two forms, D form is 75 times more potent than the L form. The in vivo triphosphates of D and plus L carbovir, they are equally active, whereas the D form without phosphate is 75 times more potent. This is again a reverse transcriptase inhibitor. Here also it will incorporate into the viral DNA and disturb the viral replication. So, but it will not have any interaction with the host cell DNA polymerase alpha, beta, or gamma. But because of the low aqueous solubility and poor oral bioavailability, as well as inefficient central nervous system penetration, this is prevented from further developing as anti-HIV agent. So this was originally synthesized from vincilactum. This is vincilactum, which is a racemic mixture. This can be resoluted into its nanomeric forms through Pseudomonas fluoresceum, or this can be hydrolyzed through a esterification into this ester amide in the form of racemic mixture. Then it can be resoluted through pig liver esterase. So depending upon any of the methods used, then through reduction, this can undergo reduction with lithium borohydride to give us the corresponding alcohol and amine, and then this will condense with 2,6-diamino purine to give us this type of racemic mixture, purine derivative. Then this can be resoluted through ADA enzyme to convert into carbon. So this is how we can have carbohydrate. Then another NHU selective synthesis was reported by Jones and co-worker from Glaxo Industries. So what they have done is they have started from this chiral epoxide, and they have developed two different routes. First route is that if we react this with PMB, paramethoxybenzyl chloride, in sodium hydride, then the corresponding ester will be formed at this place. Benzyl ester, methyl ether will be formed. When this is reacted with this base in presence of triphenylphosphine and iode, we will have the ring opening. This will attack at this position, and this will become OH, as you can see over here. Then this reacts with this thiochloride derivative in presence of DMAP and tributyltin hydrate dried in azabis isobutyronitrile, and basically this hydroxyl group will be replaced by hydrogen because of this tributyl tin hydride. So first this will oxidize this into the corresponding ester, and then that ester in presence of tributyl tin hydride through uh, free radical reaction will convert this into alkane. So alcohols, they are converted into alkanes by this method. Then through DDQ, the hydrolysis of this ether linkage will take place, and we will get the corresponding alcohol, and then dehydration will take place at this position in presence of acetyl chloride, DMAP, and this base. Then hydrolysis of this benzoate in presence of base will take place, and we will get carbamide as the product. Second method is you react this with acetyl chloride, DMAPTF. Directly dehydration will take place. We will get this carbon-carbon double bond. Then this will react with this base. This nitrogen will attack the epoxide, as you have seen in the first method also. We will get this type of compound, and then this hydroxyl group will be converted into the alkane, as you have seen in this step. Same way, Then this in presence of boron ethate, acetic anhydride benzoate will be converted into acetate. And here also N-acetate will be formed. Then this will react with NaNO2 acetic acid. This amino group will be converted into keto group, and then hydrolysis of this acetate and this N-acetate will lead to the formation of carbon. Then this is the latest method which is developed by Rera and co-workers, and different substrates they have been used. This Boston Kant PK adduct A. This is a chiral cyclopentanol. This is used as a substrate, and this is synthesized from this bicyclic compound uh, from trimethylsilyl and norborandine, this one. So when they condense with each other, cycloaddition reaction, they will give us this Poisson-Kant PK or PK adduct. When this adduct is reacted under these conditions in presence of KCN ammonium chloride DMF followed by reaction with the 2,2-dimethyl 1,3-propane diol, then protection of this keto group will take place. KCN will add to this double bond, cyanide will be formed, and protection of this ketone through 2,2-dimethyl 1,3-propane diol will take place. Then on reaction with diol followed by sodium borohydride, this nitrile will be converted into amide first, and then that amide will be reduced to uh, the corresponding alcohol, and on hydrolysis you will get alcohol, and then tips Cl will give you this OTF, O-tips derivative, and then aluminium dichloromethane will uh, give us conversion of this into alpha beta unsaturated ketone with the removal of the other ring. Then this will react with the diol THF. Diol will selectively reduce ketone function group and will not affect carbon-carbon double bond. We will get it converted into alcohol, and then that alcohol will react with ethyl chloroformate, and this ester will be formed. Then this ester will react with this base, then this nitrogen will attack this carbon. With the removal of this group, we will get this type of intermediate. So this intermediate is then converted into an isomerically pure carbamide when this is reacted first with TBF in THF at room temperature. Hydrolysis of this protection group will take place. This alcohol is formed, and then likewise on reaction with sodium hydroxide, this chloro will be converted into ketone, and we will get carbamide as the product. So these are the references which have been used in preparing this lecture. Thank you very much.